Okay, hi everyone. Thanks very much for inviting me to talk today. I'm a specialty registrar in clinical pharmacology and an inaugural Health Education England Genomics Education Programme fellow. And I'm going to be talking about precision dosing and pharmacogenomics. So on the front line, it is very clear that there is widespread variation between patients in how they respond to a given drug, affecting both the level of efficacy attained and the occurrence of adverse drug reactions. We've already heard that this is clinically important. It's estimated that 6 to 7% of all hospitalizations are attributable to an adverse drug reaction, 15% of inpatients experience one. Moreover, it was estimated that only half to 3/4 of patients respond beneficially to first-line therapy across a spectrum of therapeutic areas. Therefore, variable drug response negatively impacts patient outcomes, and patient safety, and the quality of care that we provide. Given that it's projected that global annual spending on medicines is going to exceed $1.4 trillion in just 2 years' time, this inefficient and ineffective use of drugs will carry a substantial potential cost burden. When I think about prescribing in practice on the ward, it often seems to me to occur right at the end of the patient journey, and as a bit of an afterthought. The patient is presented with their symptoms, they've gone through the history, examination, investigations, all focused quite appropriately on making the diagnosis. But then the pressed physician has to prescribe a dose, we do it quickly, and often it's done in a sort of one-size-fits-all paradigm. What's the effect of this? Well, when you then measure levels of a given drug in a cohort of patients, you find that there's substantial variation in the level detected, and there'll be patients when it's known both in the subtherapeutic range and in the supertherapeutic range. And indeed, then when you survey the academic literature across a range of commonly used drugs, it's apparent that there's substantial fold changes in both the concentrations measured and in the concentration time exposures measured between patients, although the heterogeneity between different academic studies means that it's quite difficult to determine between drugs which has the greater variability. But overall, there appears to be substantial pharmacokinetic variability. But that's not all. There's also variability in the sensitivity of the on and the off targets to a given drug. In other words, a given concentration of a given drug can have different effects in different people. There's multiple factors that are responsible for causing this variability, not least lifestyle choices such as smoking, environmental exposure such as concomitant drugs and foodstuffs, comorbidities, genomic variation, and other omics such as the epigenomics, metabolomics. Therefore, I'm hopeful that in my clinical practice we're going to move towards a more intelligent, integrated system whereby the factors which are affecting drug response are collected earlier, right at the start of the patient journey. They are integrated, they are stored for future healthcare interactions, they can be provided to the patient when they want them, and then when the pressed healthcare prescriber is forced to come up with the management decision, they've got recommendations being automatically generated to help pick the drug and the dose most likely to help that given patient, and subsequent rapid review to assess the effects and optimize our management. Why do I think pharmacogenomics is part of this, as well as drug and food interactions? Well, for several reasons, but not least that it's estimated that over 95% of the population carry at least one high-risk genotype within established pharmacogenes which have been related to differential drug exposure. So on that sort of backdrop, I now want to use a few different drugs, warfarin, statins, and touch on alugastatin rare variation as vehicles to further explore the pharmacogenomics of precision dosing. So first up, warfarin. It's still the most commonly used oral anticoagulant. About 1% of the UK population are on it, and there's a 40-fold variability in the doses prescribed. And it still remains the third most common culprit drug responsible for an adverse drug reaction leading to hospitalization. Warfarin is a vitamin K antagonist. It's a racemic mixture of two enantiomers, and they specifically inhibit the protein vitamin K epoxide reductase complex 1 within the liver, and this reduces the activational post-translational gamma carboxylation of clotting factors 2, 7, 9, and 10, as well as proteins C, S, and Z. The S-warfarin antimer is about 3 to 5 times more potent at inhibiting VCORC1, and it is metabolized predominantly by the 2C9 isoform of cytochrome P450. This is one prospective UK-based study which carried out a genome-wide association study on warfarin mean weekly dose and found 2 very strong genome-significant hits in both the pharmacodynamic target, VCORC1, and CYP2C9. And in VCORC1, it's a common variant. It's in the promoter region, so it doesn't change the protein sequence, but it's associated with reduced transcription of VCORC1, leading to greater sensitivity to warfarin. And that can explain by itself 25% of all the variability seen between patients in their warfarin dose. CYP2C9, the 2 more common haplotypes in Caucasian star 2 and star 3 are also responsible for about another 15% of observed variability. And there's a cluster of other clinical factors which further contribute to explain variability. So overall, we can explain about 50 to 60% of the variability we see in warfarin doses in our patients today. And this has led on to a number of randomized controlled trials, 3 seminal trials, 2 of which were published simultaneously. UPACT in the UK and Sweden looked at this thing called time in the therapeutic range, whereby it's the amount of time patients are in a therapeutic level of anticoagulation. So we measure the international normalized ratio, and you want patients generally to be between about 2 and 3. And they found that taking into account genetic information significantly increases the time that patients are in this therapeutic range from 60% to 67%. But at the same time, the COAG study based in the States found no significant difference between pharmacogenetic and clinically guided dosing. Just published most recently from the States last year was the largest study to date, which was actually powered for a composite clinical endpoint, 30-day risk of major bleeding, INR greater than 4, death or venous thromboembolism up to 60 days. And they also found that taking into account genetic factors led to a significant reduced risk of this composite clinical output. So overall, there is the thrust of the evidence is favour of precision dosing of warfarin, taking into account these pharmacogenetic variants. Just to go into a little bit more detail, it seemed that there was an early separation in patients starting warfarin in the UPAC study, gaining benefit from pharmacogenetic prescribing. And importantly, compared to the controls, they reduced this initial spike in a high INR, which would be associated with a greater risk of bleeding. Who benefits? So which patient subgroups benefits? Well, it transpires. It's the patients who are carrying the greater burden of the variant alleles in CYP2C9 and VCORC1. And those patients with 2 or more variant alleles actually experienced the benefit of 11% difference compared to their peers in the control group. And that's a clinically significant and relevant absolute difference. And this, from the UPAC study, this pharmacogenetic approach was shown to be cost-effective as well, which is important as we're here to talk about implementation. So why there was a null or a negative trial result from COAG? There's been lots of postulations about this. I'll just touch on one observation, which is that the patients recruited to that study were ethnically more diverse and included about 27% of patients of black ethnicity. And however, the main genotypes taken into account were stars 2 and 3, which are more prevalent in Caucasians than in those of black ethnicity. However, those of black ethnicity have a higher frequency of other reduction of function CYP2C alleles, 5, 6, 8, and 11, which were not taken into account. And this could lead to misattribution. So you would think they are wild type, but actually they carried one of these reduction of function variants, which would have made them more sensitive to warfarin. That's just one of the postulations. Clearly, warfarin isn't the only player in the anticoagulation field. There's also the development of the direct oral anticoagulants. And I'm just going to touch on an idea that, because they're still on patent, they're very expensive, perhaps stratification might be a way forward. Because again, if you take from these randomized trials, if you stratify patients based on the number of variant alleles in VCORC1 and CYP2C9 which they carry, When patients don't have any of them, they actually don't experience any increased risk of bleeding compared to edoxaban. But it's the patients who are on 2 or more, these highly sensitive people to warfarin, who actually experience a benefit in terms of reduced risk of bleeding on edoxaban. And so you can envisage a mechanism by where we genotype people, and if they're all wild type, they can safely have warfarin, and if not, perhaps then we give them the more expensive DOAC. And similarly, when this meta-analysis looked at time in the therapeutic range, and for a site, and when that time in the therapeutic range goes above 66%, there's no difference in bleeding risk between DOACs and warfarin. But when that time in the therapeutic range for warfarin drops, then suddenly DOACs become more beneficial. So that's warfarin, it's the longest. I'll just mention statins now. These are one of the world's most commonly prescribed classes of drugs. They're very effective at reducing cardiovascular events, but they're associated with some adverse drug reactions, muscle toxicity and incident diabetes. The muscle toxicity occurs on a spectrum from the more common, non-specific benign myalgias through varying degrees of severity of myopathy with elevated plasma creatine kinases to severe rhabdomyolysis with associated acute kidney injury, and a rare, relatively new entity of an autoimmune necrotizing statin-mediated pathology, which is associated with autoantibodies to HMG-CoA reductase, which is the pharmacodynamic target of statins. Focusing though on the more myopathies, the rhabdomyolysis things, clinical factors have been associated with an increased risk, other drugs, lower BMI, older age, female sex, and untreated hypothyroidism. But a seminal GWAS came out in 2008 from the Search Consortium, which looked at patients, first of all, on a very high dose of simvastatin, 80 milligrams daily, compared to controls, and they found a single genome-wide hit in this gene, SLCO1B1. That variant was in strong linkage disequilibrium with a non-synonymous variant within this gene, which increased the risk per allele by 4.5. And actually, if you were homozygous recessive for this non-synonymous variant, actually the odds ratio was 16.9. And they furthermore replicated this in the heart protection study with a lower dose. Patients were on just 40 of simvastatin, and it had a corresponding lower risk estimate. SLCO1B1 encodes organic anion transporting polypeptide 1B1. This is a transporter in the liver. It's an influx transporter, and healthy volunteer studies have shown that as you have the recessive alleles, that's associated with an increased plasma exposure of simvastatin acid, the main metabolite of simvastatin. But it's also actually associated with an increased exposure to all statins, apart from fluvastatin. And this has led to changes in clinical practice. The FDA put out a safety communication recommending not to start people on 80 milligrams simvastatin unless they've been tolerant to it for 12 months. Over in the UK, the European Commission's summary of product characteristics recommended that if the genotype's known and people are homozygous recessive, then perhaps you should give a lower dose of simvastatin. Some work I've been doing looking at the prevalence, predictors and prognosis of something I'm interested in, suboptimal statin therapy, and I just wanted to touch on this. We've been carrying out a secondary prevention cardiovascular cohort study within the UK that recruited 1,470 patients admitted to one of 16 hospitals with a non-ST elevation acute coronary syndrome. And I was interested in looking at those patients who were discharged on a very high dose of statin, almost exclusively atorvastatin 80 milligrams daily. And I was surprised to find that by just 1 month follow-up in the community, 15% of these patients, they've either stopped their statin already, having just had a heart attack, they're non-adherent to it, or their dose has been reduced or been switched to a lower statin, another statin of lower equivalent dose. And when I wanted to look at factors that might be associated with this, despite the low absolute numbers, muscle symptoms which were reported by the patients were associated with an increased risk of this composite of suboptimal atorvastatin. And when I wanted to look at the prognosis, well, in multivariable adjusted analysis, time to both all-cause mortality and major adverse cardiovascular events, namely another heart attack or a stroke or a cardiovascular-related death, there was an increased risk of these people identified just 1 month after a heart attack as having suboptimal statin therapy. So then I wanted to explore a little bit further, so I went on and carried out a pharmacogenetic study, measured the levels of atorvastatin and its metabolites in 590 of these patients, And although it doesn't hit genome-wide significance, but that non-synonymous variant right back in SLC01B1 also flagged up here as being associated with increased atorvastatin in this large cohort of patients. And then furthermore, then I put this variable back into my model and found actually it's also associated with this increased risk of suboptimal atorvastatin therapy. And so perhaps I posit in patients that carry this allele, perhaps they should go on a slightly lower dose of atorvastatin, given that it's going to have an equivalent exposure, and that may improve their long-term adherence to this drug, which may affect cardiovascular outcomes. We'll see. I've also, this is unpublished, but I've had some very strong genome-wide hits, but there's more work to be done there. So very briefly, those are existing drugs. There's a new drug, elugostat, which came out just a couple of years ago for this rare lysosomal storage disorder, Gaucher's disease, which is where it's homozygous recessive for an enzyme glucocerebrosidase, and that leads to an accumulation of its substrate, glycosylceramide, in cells of particularly the monocyte macrophage lineage, which then leads to infiltration in the bone marrow and hepatosplenomegaly, and this leads to sequelae including anemia, thrombocytopenia, bone pain, osteoporosis and fractures. Previously they had to be on enzyme replacement therapy, which is intravenous. This elugostat is an oral drug, and it's a specific potent inhibitor for the enzyme that works in the other direction, glycosylceramide synthase, to reduce, it's called a substrate reduction therapy, reduce the amount of glycosylceramide. And it does, it decreases spleen volume, liver volume in these patients, but it is associated potentially with prolongation of QTc interval. We know that it's metabolized mainly by CYP2D6, and therefore for a newly licensed drug, it was recommended based on physiologically based PK monitoring that those who are poor metabolizers on a genetic test in CYP2D6 have half the dose of those who are extensive or intermediate metabolizers. Furthermore, in the product's labeling, they've actually gone into drug-drug gene interaction, so thinking about patients who are also on concomitant CYP2D6 or 3A4 inhibitors. So that's, I think, a bit of a vision of where we want to be heading, integrating it all together. Very lastly, it's clear to me that we've got this ever-expanding list of drug gene associations, but the variants in those genes are generally common. However, when people have done an exome sequencing analysis, in the exomes at least, the vast majority of variants are rare. They're population specific, and in the pharmacokinetic related genes, it's been estimated that 30 to 40% of their functional variability may be due to rare variation, which we're not considering at the moment. And given that the vast majority of adverse drug reactions are type A or exposure related, this could be important to our patients. This is just to give a little outline of the fact that the proportion of predicted functional variability in our pharmacogenes, which are rare or common, does vary on the, as you might expect, on the particular gene. So do we know anything about the functional impact or clinical impact of these variants? Well, I think this is really in its infancy, but one study found that methotrexate levels are also associated with that same transporter as simvastatin, SLC01B1, and of the proportion of variation in drug level attributable to SLC01B1, 18% of that was due to rare non-synonymous variants. P450 oxidoreductase is a gene which is an obligate electron transporter. It's needed for all of our CYP enzymes to actually function. People forget that it's very important, and a rare variant, A287P, which in people who are homozygous recessive for that actually presents with a rare disease, whereas the heterozygous form is asymptomatic. One study taking a patient and her asymptomatic heterozygous mother found that compared to population expected levels for a range of CYP genes, they were both had very low levels of activity for most of the genes, although actually the mother seemed to have relatively good CYP3A4 function. Whole exome sequencing has shown that there's rare variants in CYP3A4 for paclitaxel. And finally, there seems to be in patients who have drug-induced long QT syndrome a prevalence of rare variants, amino acid coding variants in the congenital long QT syndrome genes. So this has led on, I'm commencing my fellowship with the 100,000 Genomes Project, which is a world-leading resource, in order to try and determine some of this clinical utility and the potential health economic benefit of considering the full range of variation, both common and rare, in pharmacogenes. And it has 4 work packages. The first one, which I'm undertaking at the moment, involves collating a list of pharmacogenes and then cataloguing all the variation we find in it. The second is then carrying out in silico analysis to predict the potential effects of these functional variation. The third, the most complicated, is then to carry out some specific case control studies where I'm going to see whether this variation is associated with specific adverse drug reactions to a few different drugs, and finally look at some potential cost-consequence analyses of these associations. So thanks very much for listening, and for anyone who likes acronyms, I'll leave you with that. Thank you.